Hey, that's not me. That's somebody else. Where's, sorry, folks.
It's okay. I bet we could scroll through a lot of these though.
We sure, yeah, they're all good. Here we go. Can you hear me?
Yes.
As a biologist studying tropical rainforests, my primary focus was on doing the science itself. But I quickly learned that it wouldn't be possible to sustain that work unless we partnered with communities to find solutions to the challenges that they're facing. One can't work in a place like New Guinea without developing a partnership in which there really is reciprocity and sharing of knowledge and sharing of benefits. As a curator in a natural history museum, we discover new species, but I have to put the species discovery in quotes because the people with which I work take these plants for granted. They are only discovered in the sense that they were not known scientifically, but they might have been a very important part of cultural practice or ways of living. The communities in which I work increasingly recognize how threatened their cultural resources are. The extinction of languages and this indigenous knowledge is happening faster than we're losing biological species. So for me, a real shift in my work, kind of a pivot from doing some basic science has been to partner with communities that wish to record for the first time some of their oral history, their languages, their names and uses of their plants and animals. Last year, it was an honor to be a part of a launching of a guidebook to indigenous knowledge and biodiversity for a particular community that has dedicated its own conservation area. Papua New Guinea may be halfway around the world. It might be just about as different an environment as you can possibly imagine, and yet so many of the challenges that we face here are shared there, and there's some insight on both sides to be gained. My students going abroad to Papua New Guinea have had experiences like my own that have changed our lives and our outlook forever. Now we are embarking on training the second and third generation of these indigenous scholars, these citizen scientists who have translated their traditional ecological knowledge into the language that we speak using the technology of the 21st century. It's pretty amazing.
Okay. So that was one little intro. And now turning to cannabis. I've used kind of a lot of the same approaches in the cannabis work that folks in New Guinea taught me. And it was, I think, Clement Dabney, who's here, also was doing his PhD at the time when Fox 9 News decided to do a piece on our cannabis research. And it was at the time when the hemp-derived THC was just hitting the marketplace. And so they did this little story. I'll show that next. Are we good on the audio and video on that last piece that came through? Okay. Okay, great. All right, here, let's see if I can dig this up. Just give me a moment. Okie doke. I think I got it.
Well, maybe you've heard your teenagers or maybe even some friends talking about something called Delta 8.
This is the hottest new cannabis product. Some are even calling it Weed Light. Here's Tom Lydon of the Fox 9 investigators.
It began showing up a few months ago in smoke shops around Minnesota.
Yeah, definitely. It's popular. Yeah. In fact, I'm kind of low on stuff, actually.
And selling out fast.
You see, I'm almost sold out.
Oh, yeah.
People, I don't know, they are coming by 2, 3, 4. What the?
It's called Delta 8. It comes in gummies, vapes, and flower or bud.
Yeah, you can smoke it. Smell it.
Oh, yeah, that's.
It's good stuff, yeah.
The store clerks will tell you it's got a little more kick than CBD, but not quite the punch.
And how high does it get you? I mean, it's half the strength of like regular THC, I would say.
And the clerks will tell you something else. It's all perfectly legal.
Is that legal to sell? Yep, this is 100% legal.
You sure won't find much information on the labels. So we took a couple of samples to a lab for testing. We'll tell you the results in a moment. But first, a little Cannabis 101. Your professor, U of M botanist George Weiblen.
What this does is tie together all these different families of plants to a common ancestor.
Weiblen's lab recently mapped the cannabis genome and discovered marijuana and its country cousin hemp actually share about 90% of the same DNA. They also share most of the same chemicals, 144 so-called cannabinoids like CBD for what ails you and THC to get high. Its scientific name is actually Delta-9 THC. And that's important because come to find out there's a nearly identical molecule found in much, much smaller quantities called Delta-8. It has some of the same intoxicating effects as Delta-9. It's just less potent, earning the nickname Weed Light.
And they hit the same receptors in the brain, correct?
Right. Yes, they bind to the same endocannabinoid receptors that occur in our central nervous system.
It sort of looks like a duck, quacks like a duck, and acts like a duck in terms of being very much like marijuana.
Yeah, it's a duck.
Maybe a duck that's only half-baked. Problem is, cannabis doesn't produce very much Delta-8 naturally. So while it can be extracted from legal hemp, it's more commonly synthesized from CBD through a chemical process known as isomerization. In many cases, Delta-8 may actually be sprayed onto the hemp flower.
Spiked weed, basically.
As a botanist, Weiblen is downright offended by the bait and switch.
It's exploiting the industrial hemp legislation. It's like, look, if you're going to grow drugs and sell drugs, let's call them drugs.
The farm bill that legalized hemp doesn't mention Delta-8, but it makes hemp products legal as long as they contain less than 0.3% of Delta-9 THC. But then last August, the Drug Enforcement Administration said the farm bill doesn't cover synthetically derived tetrahydrocannabinols that don't come directly from the hemp plant itself. In that case, Delta-8 would fall under the Controlled Substances Act, just like Delta-9 THC. In other words, Delta-8 is legal if extracted directly from hemp, but maybe not if synthesized from CBD, even if that CBD was originally extracted from hemp. Confused? You're not alone.
I think Congress is going to need to act. Congress is going to have to decide once and for all how to regulate the plant cannabis sativa in all of its forms and varieties.
Cody Weiberg's executive director for the Minnesota Board of Pharmacy. The picture is just as confusing, he says, under state law, which allows the sale of non-intoxicating cannabinoids from hemp. Problem is, Delta-8 is intoxicating. Another state law makes it illegal to sell a misbranded drug not approved by the FDA. And clearly, people are making medical claims about Delta-8.
Oh, what kind of pain relief?
Just pain relief in general.
How about anxiety?
All of that. Inflammation, anxiety, all of it. It's going to help with all of it.
If there's no advertising, nothing on the label, it's harder for regulators to prove intent.
I think it speaks also to just the state that we're in with COVID-19. We're seeing absolute skyrocketing of mental health issues. And I think many of us are desperate to get advice or some relief anywhere that we can. And
Dr. Stephen Dahmer is a medical director for Green Goods, one of two licensed producers of medical cannabis in Minnesota. He worries Delta 8 could set the entire cannabis industry back.
It's buyer beware that we don't know what we're getting, and we don't know what else might be included in that.
Which brings us back to the samples we took to Legend Technical Services in St. Paul, which does testing for the hemp industry and is one of the few labs we found that could test for both Delta 8 and Delta 9 THC. The flower, or bud, is nearly 2% Delta 8, more than the plant would make naturally, meaning it could be spiked according to our experts. It's 14% CBD, that's quite a bit, and twice the legal limit for Delta 9 total THC at 0.75%, but likely not enough to be intoxicating. The gummies, on the other hand, are 0.6% Delta 8 and just minuscule amounts of THC and CBD. Keep in mind, you wouldn't know any of this looking at the labels.
How is it going to be regulated? And there needs to be some sort of national standard.
Because standards and regulations, as uncool as that sounds, is how we keep consumers safe in America.
For those of us who put our trust in evidence and evidence-based solutions, this is a nightmare.
Weiblen wants us to wake up and realize it's not two plants, marijuana and hemp. Genetically, it's just cannabis in all its varieties. And if we're serious about its perils and promises, maybe it's time our laws were too. For the Fox 9 investigators, I'm Tom Lydon.
Okay. Well, curious to get any questions or feedback about either of those pieces. See if there's anything in the chat. I tried that Delta stuff and it made me so sick.
Well, I think it's good news.
What is good news?
Well, I think it's good news about that it helps with inflammation and it kind of the after effects of the whole pandemic shutdown thing and the fear that's around. And so I think if there's a way that people can begin to be more social and work together on things and stuff like that, that's totally essential for us in where we're at with the planet, you know. So anyway, that part is good news, and it helps with inflammation. And so
yeah, that's a really that's a really interesting point. You know, you could see from that second video that it was done during the pandemic, and it's sort of everything, you know, is sort of permeated with fear, you know, and this idea that we need to be taken care of, and we were all isolated, and you know, and now we've come out of that, and we can come together and work on things together, and move, you know, to affect, you know, positive changes, and that's what excites me. That's what excites me about working on hemp. It's really challenging. It's really challenging. I really am driven, you know, more than anything else to move it in the direction of weaning our addiction to fossil fuel based plastics and fossil fuel based materials and fossil fuel based industries. And so it's that industrial hemp piece that really has motivated me for a long time. But then, of course, we have the cannabis as medicine part of it too. And that's an important part. It can't be ignored, but it complicates my life when I'm trying to focus on hemp for fiber and materials and things like that. Could someone tell me a little bit about the most recent topics y'all have covered when you have gathered recently?
I was only at a few of them, but I mean, I was only at some of them, but we talked about, hemp construction. We had, Cameron and Danny Desjardins on talking about the hempcrete houses over there. We had, the bioremediation folks on last week to talk about some of the potential bioremediation. They were up in Maine and they've been doing some work on a military base up there, I think, that has whatever nasty things the military leaves out there. We had our growers on explaining how to grow it because, you know, some people are like, oh, you need a planter. I was like, yeah, you need a seeder. You know, we were talking mostly about industrial hemp, right?
Yeah.
And we talked, you know, and then we talked kind of about our work at Winona's Hemp and AAI. But and I'm not sure if we had another one on the genetics. I don't think we did, but that was kind of you. And then I thought I saw Clemen's incredibly ornate charts on genetics. And so you know, I figured you guys could explain something on the family
Yeah.
and you know, the different kinds in the, you know, in the industry. Oh, and then Ira talked about the indigenous standard one day, I believe, right? And their work down there to just give a sense of like what it is like to be artisans that then incorporate hemp into your artisan work, and you know, just a really beautiful amount of work that they're doing down there. So it's going to be kind of a range. You know, still upcoming is hemp batteries, you know, and supercapacitors.
Okay.
We have a workshop of that. So the intention here is really
Cool.
just to give a broad sense, and I think we'll have one more on textiles, a broad sense on, wow, look at what you could do with hemp, and just to, you know, inform people. A lot of tribal governments are looking at it, but they, you know, some are mostly just marijuana, but it's so, you know, there's so many things. So
Yeah.
there's a little bit of a sense, George. I don't know if anybody else
Yeah.
wanted to add in there on what there was, but yeah.
Well, thanks, Winona. I also see, you know, Esther put in the chat encouraging me to talk a little bit about what I've been through to set up the hemp work at a university to work, and then the work we've done with the Anishinaabe Agriculture Institute and also with Sisseton and Wapatanoyate. So I can roll with that. Yeah, other comments, questions before
Yeah,
I.
talk about your pursuit of the feral hemp. I was
Yeah.
telling them about that.
Yeah.
George is wandering around for the feral hemp, but then we got to make sure that Clement also could talk in about the varietals. And I don't know, you know,
Yeah.
if you want to talk first for a bit and then put Clement on to talk about. Were you going to talk mostly about varietals, Clement, or your industry business?
Sure, I can talk about whatever.
Yeah. Well, yeah,
These are the
I'll
two
kick.
premier guys in Minnesota. That's what I just want everybody to say. So if you got questions,
I'll
make
kick
sure you
off
can
and then
ask them. But these are the guys.
okay, I'll kick off and then hand off to Clement in about 20 30 minutes. How's that sound?
Perfect.
Okay, cool. All right. Well, yeah. I'm gonna jump back to the slide deck here. And where did that go? Share screen. There it is. Yeah. So, well, over 20 years ago, so now I got hired as a young professor to teach and do research on plants at the University of Minnesota. And, you know, one of the things that it's been very proud of is that it's like the premier land grant university and prestigious and doing all this amazing work. Well, you know, it's been brought to the university's attention that, in fact, this land grant status involves a whole lot of indigenous resources that have supported the enterprise, you know, since it was kicked off back in the 1870s. And there's a lot of work that needs to be done to repair relationships with indigenous communities and to return and share resources. I wasn't really aware of this when I started 20 years ago, but it turns out through the hemp work, it's kind of come to my attention, and it started to become a part of what I'm fighting for and working on within the university. And it's really a privilege to be able to bring bring indigenous perspectives into the way that agricultural research is can be done. So with the hemp, it's actually, in the work with Clemen, we are we're doing things that turn out to be a little bit unconventional for professors doing, agricultural research, mostly funded by big ag. Universities recently started to work on some new policies around uh rec- you know recognizing and healing relationships between universities and native sovereign nations and indigenous people. It turns out the hemp work uh in the ag space may be one of the first attempts to actually do some of this. So I'll tell you a little bit about, my work at the U and how that got started, and then give some of these examples. I'll pop out and let Clement talk about some of the work he's been involved with in a little bit. But I'll start with just a few clarifications I think are important when we're talking about cannabis. We can kind of think about the different uses and the different products, as we've just heard. The piece we just listened to was about, cannabinoids and their use as drugs and medicines. My work has been primarily over in the other space here, looking at hemp um, as as a source of grain for its uh, good quality fat content and protein content in the grain that can be such a healthy source of nutrition and also body care, skin care materials. Um, that's a very that's a seed that's associated with the nutritious seeds of hemp. Um, so much loved by birds in the wild. The birds know where to get good nutrition, and they go for hemp seed. It's the other stream here under hemp, the stalks, with their fibrous quality. The hemp plant's closest relatives are other plants that have really fibrous stems. Things like elm trees, nettles. Uh, these are plants that if you try to pull apart the stems and twigs, you can't break them because they've got this very fibrous outer sheath, and I call that the bast fiber. But then the inner core, this pithy core that we call the hurd, when those materials are separated, the bast fiber from the hurd, they find really different uses. The hempcrete relies a lot on the hurd material because it's lightweight, and has some antimicrobial properties and has good insulating properties. The bast fiber, has got such great natural strength that's what you're looking at uh, for fabrics, rope, for things like geotextiles, these woven mats that can be used to cover and aid in erosion control of disturbed ground and in restoration of landscapes. Um, so working primarily in this hemp space, I want to distinguish like hemp as a crop from drug cannabis as a crop. And the way I like to separate them is in that when you're growing grain or you're growing a plant for fiber use, you're going after large volume, big areas. It's a big acreage field crop. It's grown, it's grown in the field. Whereas drug cannabis, these are carefully tended, manicured plants that are produced by highly intensive horticulture on a small scale. These are really, it's like the distinction between um, growing a commercial crop as a farmer or growing cut flowers as a commercial supplier of the cut flower trade. That's really the distinction between drug type and hemp type cannabis in terms of how they're produced. We see a lot of confusion as people talk about cannabis, sort of drug cannabis growers who want to expand beyond the hothouse or beyond the greenhouse and grow in a field setting. It just doesn't work. It's just too labor intensive. Same time, you can't get enough fiber and grain out of a highly intensive horticultural operation. You need to be thinking acres to do it. Our basic work getting started, we had to go through all the hoops of getting approval from the DEA to do research at the university. The university is bound by, state and federal regulations, gets a lot of state and federal funding, and the number one issue being, compliance. So I was discouraged as a young professor at the university from going down this path, just because it was so damn difficult. It's like you're gonna, nobody gets these permits. Turned out that only one other researcher at a university in the country had the permit when I got my permit back in 2002. It took a lot of patience, a lot of persistence, and also a willingness to do a whole lot of things that didn't make any sense, like invest in security systems to, protect the laboratory from potential break-ins. Well, who would want to break into the lab and steal my hemp anyway? Because it's no good for getting high. Anyway, we jumped through all these hoops and established this lab in order to try to understand how genetically hemp and marijuana come to have such different levels of THC and CBD, these two major cannabinoids. And what we did was really the classical kind of genetics experiment where you take two different plants and you breed them together, and then you look at the inheritance of their characteristics among the grandchildren. And that's what we did. We hybridized hemp and marijuana and then grew out the grandchildren from that cross, and measured their cannabinoids and looked at their DNA. That was really that was the work that we originally got permission to do. And then starting in 2014, with a change in federal law that opened up industrial hemp for research, we were able to then come out of our highly regulated and highly locked down laboratory and start to think about doing stuff in the field. And so that the first work we did, once we were able to get out from underneath just the restricted work that the DEA permitted, we were able, under Minnesota's law, to be one of the first 5 holders of a license to do industrial hemp in Minnesota. Winona and Don being one of the other license holders. And we got so a little bit of money from the Minnesota Department of Agriculture to test hemp grain and see how different varieties would grow. The study originally was just going to include university experiment stations, but we were contacted by White Earth Natural Resource Department and asked, "Hey, could we join in the study?" And we said, "Well, that'd be great, but we may not have the money. Let us go back to Department of Ag." We went back to Department of Ag. They provided additional monies to be able to include White Earth in the grain variety trial, which was a an indication that, hey, you know, the state of Minnesota is willing to support partnerships, and we're still continuing with that kind of support even now this year. So what we did back in 2017, first time we could ever grow cannabis legally outside in Minnesota, we grew out 12 different varieties. And in the picture here, you can see the St. Paul campus. This is the Minnesota Agricultural Experiment Station. The water tower for the campus is over here. And we were able to grow out 12 grain varieties that we imported from Canada and Europe. And this is an aerial photo taken with a drone of these small 15 by 30 foot or 10 by 30 foot little patches of all the 12 different varieties. And we were interested in knowing, you know, how much grain could you produce, and what practices would you need to use to produce hemp in the field in Minnesota. And our partnership with White Earth Natural Resources, we learned a really interesting thing, and it wasn't by design. It was just something that emerged naturally from the nature of working with partners and honoring the partners' input on how we do the work. The land at Callaway, Minnesota, that was planted out with this hemp was tribal land that had been leased for corn and soybean production for a long time. It was soil that had been abused with the sort of conventional high fertilizer, herbicide, and, you know, fossil fuel input kind of agriculture. And the folks when this the folks at White Earth Natural Resources wanted to move away from that and not use fertilizer. The ag experiment station at the university, they use all the chemicals, they use all that stuff, except for the organic agriculture program. It was just given that, you know, they were going to apply fertilizer. But at White Earth, we agreed not to do that. It didn't happen. And what we noticed was that the plants at White Earth were yellow and stunted compared to tall and dark green at the sites that were fertilized. And so even though it wasn't part of our experimental design initially, it taught us that, you know, hemp really is a very nitrogen demanding crop. It needs about as much nitrogen as corn. And you know, when we started this work, it was a lot of folks were assuming, no, hemp does does really well in the wild on its own. There's ditchweed all over the place. Turns out now we know ditchweed isn't all over the place. Ditchweed tends to thrive in places where there's a lot of nitrogen, like in pastures, around livestock. Um, Along the sides of agricultural fields that are getting over fertilized. This is where hemp thrives. That's where ditchweed grows. And you give it enough nitrogen, and it grows really well. So, we learned from this, from uh, this first season of collaboration that hemp does need you need to be concerned with the nutrient inputs. We also learned that hemp is vulnerable to competition from weeds. If you don't if you don't plant at the right time and in the right density, competing weeds are a real problem. But if you provide the right conditions, and you are able to plant into a field that's relatively weed free, and you get a lot of strong establishment of the hemp, once the hemp comes up, it will provide such dense shade that it can effectively suppress any nearby weeds. So just learning about these conditions under which hemp can thrive was some of our initial work.
Could I
Um,
ask a question?
yes, yes, please.
Okay, does it outcompete garlic mustard? Or has any research been done on that? This is an invasive that is around
Yeah,
here that's
it's
really
huge.
difficult.
Yeah, garlic mustard is a huge problem. It's a problem in my neighborhood. Um, you know, uh the hemp can outcompete weeds like garlic mustard if it's planted at the right time with sufficient moisture, uh and um and the right soil conditions. We know that if we know that the preparation of the soil is critical. You can't just sow the hemp over the top of a field that's full of weeds. The weeds have already gotten started and they've got to jump. But if you can till that field before you plant the hemp, you've got a chance, and you get the hemp in at the right time, you've got a chance of giving the hemp a good head start and outcompeting weeds like garlic mustard before they come back. One of the best practices we've seen for hemp with weeds, and Clement can maybe elaborate on this. Let's say you're sowing into a field that's been abandoned for a few years or hasn't been very well tended. If you can disc up that field and plow it up, uh, uh, in spring, when the soil, as soon as you can get onto that soil, then wait 3-4 weeks, let the weeds come up again, and then right before you plant the hemp, go in and plow it again to turn all those weeds over and under and turn them over good, and then throw your hemp right on top of that, ideally right before uh right before a good period of rain. And if not right before a good period of rain, then if you can irrigate over the top after planting, the hemp will come up within 6-7 days. And if it gets up that fast, and then the soil is rich, it's got enough nutrition in it, the hemp can outcompete the weeds. And that's what yeah. all too often our timing is off, or we don't put enough care into preparing the soil first, or we don't get enough moisture, and then we have a weed problem. Yeah. Does that answer your question?
Yeah, thank you.
All right. Yeah, any others?
Is it okay to fertilize with composted horse manure?
Absolutely. Absolutely. That is the best. One of the ideas around cannabis in the wild is that cannabis and hoofed animals have had a long, long history together, a history going back millions of years, uh, long before humans got interested in cannabis. This is an idea that's been tossed around by some of the experts, like Ernest Small in Canada, who's written he's written kind of the research Bible on cannabis. I'll pull it up here. Yeah, he's this thing. It's Cannabis, a Complete Guide by Ernest Small. And he's got these four things. He's got food, fiber, pharmacy, and fun. The pod pipe is the fun. Uh and Ernest Small's idea is that like in Central Asia, up that where these hooved wild animals evolved, the cannabis plant evolved with them. And the cannabis plant likes the very places where the hooved animals like to graze, which is on grasses along riverbeds, floodplains. And then where the animals like to feed, they eat up all the grasses, and they've got water there and the floodplain. Then all their manure and then all the action of their hooves is perfect for the hemp or for the cannabis to get a start, because the manure provides the nitrogen. The turning of the soil is done by the hooves of the animals. And that creates the open space where the cannabis can then grow and form a nice thick stand that outcompetes the weeds. So any kind of organic compost, but particularly animal manure is excellent. Yeah.
Yep, you got that right. That's where native cannabis is along creek beds where animals and their animal trails, and that's where you'll see the native cannabis in this area. Mhm.
Mm-hmm. Yeah, when we get out to Sisseton, Wahpeton, Lake Traverse area, we do find it in those areas like you're describing, but where we find it also just doing really well is in these feedlots where ranchers congregate all their livestock in the winter. They might have, thousands of acres that they can put their livestock out during the growing season. They move them around from field to field. But then in the wintertime, they got to bring them all together in one spot where they can feed them hay. And so these animals end up spending a whole bunch of months concentrated in one area. And those areas are just like, there's so much manure, and the ground is just mud because the animals have, and that's where that stuff really comes up well too. As super interesting working at Sisseton Wahpeton with their natural resources division. They have very, very good management of their grasslands, no overgrazing. They have very healthy native grass and wildflower communities going. And in those areas, we can never find the hemp because it can't get a start. The the plant community there is so healthy, it's resistant to any invasive species coming in. And we had to go off the tribally managed land and go on to these bachelor farmer lands. Clement, you want to speak to what that experience was like out there? I think you're muted.
There we go. I got it fixed. Yeah, so we basically had to get some permissions from some bachelor farmers, and it was just I don't even know how to explain this politely. Uh we'll just call it yeah, two older guys that
You don't have to be polite, Clement.
basically two rednecks that were missing teeth and had horrible cattle management and rotation practices. But their very poor cattle management practices allowed for them to grow quite a great feral hemp crop. And the funny thing is they kept trying to get rid of it, but they would wait for it to flower out and get seeded, and then they would cut it down, but they were just essentially spreading the seed, or they would spray it too early, and then plants would still get established, and they would still maintain populations. And so yeah, it was just like we had to go find people who were bad at farming. And we got really lucky to find some good populations there.
A question in the chat from Katie Ross. Do cattle graze on hemp? Yes, they do, but it's not preferred forage for the cattle. They would much prefer to forage on grasses, and grasses and legumes, that prairie, grasses and legumes than on the hemp. Why is it that it's not a preferred forage? I could, I could speculate a little bit about that.
It's too woody.
It's too wood. The stems, yeah, the stems are fibrous. Yep. And if you've handled this stuff, you know that it's got a scratchy texture
Right.
to the foliage and the stems.
Mhm.
That's formed at the microscopic level. These plants, just like hackberries and elms and nettles, these are formed by little crystals of calcium carbonate that the plant makes, and it protects its leaves by covering its leaves and stems with these little pointed calcium carbonate hairs, and those are irritating. I kind of have a little bit of a sensitivity to it because I've been handling cannabis so long. I get irritated, get a little bit of a rash. And I don't know, maybe that interferes with the cattle feeding on it too. Yeah, that's a great question.
Mhm.
Let's see. Has there been any long-term studies on hemp, health and production in soils that have highly functioning soil biology with natural fertilizers? How can we naturally provide nitrogen without plowing and disrupting the bacteria and fungi? Great. That's a great question too. Yeah. Clement, were you along when we visited Joe Brecker's like permaculture? What was that? Multi-crop thing that
It's basically, yeah, it was basically kind of like a multi-crop. So yeah, in between row, he was planting like nitrogen fixing crops,
yeah,
nitrogen fixing forage style crops.
yeah.
But I have not seen any long-term studies that are basically looking at no-till or even multi-cropping.
Yeah. I've heard reference to one agriculture institute that was doing some no-till studies with hemp. I can't pull the name of it off the top of my head. I'd have to dig through, but Aaron Axelrod, who'd done some work with Winona, mentioned this institute that might have done some preliminary work, but I don't think there's been nearly enough in this area, and I think it's definitely it's ripe for study. What I was asking Clement about was a farmer who's like, he's like he's a North Dakota, farmer whose grandfather was really into soil conservation before it was recognized how important it was. And this guy, although most of his operation is conventional, he is always experimenting with better ways to do agriculture for the future. And one of the things he has done now for like, 15, 20 years, he's got this field that he grows rows of corn with rows of alfalfa intermixed with nitrogen fixing alfalfa and corn. And then he's got some other native plants thrown in there too to create a multi-layered complex kind of a field. It's not just a monocrop anymore. And he's been doing the same thing. He still harvests the corn off the top every year. He doesn't get as much corn per acre from that field as he gets from monoculture. But what he's able to do now is he doesn't have to provide any fertilizer inputs to that field. He also doesn't have to provide any pesticide application to his corn. And he showed us with our own hands, he had us dig into this soil and lift it up and hold it and squeeze it and watch the worms in it. And this stuff was just, it was just like really lovely, airy, healthy soil full of microbes. And nothing like what we see here on the St. Paul campus, where the soils have been treated like nothing more than a holding bed for plants. It's just they've been abused for decades now. Just no comparison. I think we need work with hemp in a multi-crop kind of setting to really understand how this can be done. Yeah. How can we naturally provide the nitrogen without plowing? I mean, I think we're a long way from being able to do no-till hemp cultivation, but we may not be that far from doing low-till hemp cultivation. And one of the really interesting particular ideas that we haven't been able to get funding for yet would be to partner hemp with winter annual crops. So you could grow hemp with its during the short, you could grow hemp, which has a late planting date, and then you harvest it for fiber and grain in the fall. And after harvest, then sow a winter annual, like winter wheat or pennycress or camelina or something like this. Sow that in the fall to hold down the soil over the winter, and then those crops would be ready for harvest um, in in early June. And the real problem with doing this dual cropping or double cropping is that when you harvest the winter crop, it's after you harvest it, it's too late to plant something like corn or soybeans because those have an early planting date. The beauty of hemp is it has a late planting date. You can put it in after you harvest your winter annual. And so you could do that in a kind of a low till fashion, just lightly tilling the field. If the winter annual has helped keep the weed pressure down, you could do low till, put the hemp in, and then repeat in the fall after the hemp harvest, put the winter annual back in. But again, this is proposed. We don't know yet how workable it is. But it's I think it's well, it's an experiment we'd really like to do. Yeah, I see a hand up.
It would be interesting to try partnering with the Amish in way southeast Minnesota, because they're already doing it, to
Yeah.
practice intercropping with them.
Yeah, that's a really good point. There's, a lot to be learned working, with those communities
Mhm.
for sure.
I mean, yeah, you'd have to access the community, but I know them because of horses, right? You'd have to have someone that, but
Yeah.
you might be able to, they might be interested,
Mhm, mhm.
no?
Yeah. Cool. Well, it's great to have those questions. I think maybe I'll break. Can I hand over to you, Clement, to talk for a bit?
Yeah, sounds good.
All right.
Figure out how to
I'm gonna take a physiology break and be right back.
I think I just have to give Zoom preferences real quick.
That's good. This is great. These are great questions. I really like being able to kick these ideas around back and forth. It sure beats having to during the pandemic teaching on Zoom with students, and man, it's just like hard to get a conversation going. Yeah, what's your email? Put it in the chat here. All right. All right. Take it away, Clement.
All right. So yeah, I guess you guys have met me and George. Some of my background is I earned a master's in science in plant breeding and molecular genetics from the University of Minnesota, as well as a PhD in cannabis molecular genetics and genomics. I'm currently the chief science officer at the Uniflora Holistics, which owns Retro Bakery and Love is an Ingredient. Retro Bakery is an edibles manufacturing company, and Love is an Ingredient is a chain of retail stores. I am also the owner of Dr. Dabs and co-owner of A&D Genetics. So Dr. Dabs is a edibles manufacturing company, and A&D Genetics is a cannabis seed production company. And I've been a licensed hemp grower since 2018. Some background on cannabis is it was one of the first domesticated plant species. There's archaeological, and it quickly cultivated through ancient Egypt, Greece, Rome, Africa, China, and Europe. The seeds were originally used for grain in ancient China. Then there's historical evidence that stems were used for durable fiber for cloth, paper, rope, twine. And then the dried flowers were used as a recreational drug and medicine throughout the world. And as you can see on the slide here, the domestication of origin area is in Central Asia. And so Minnesota itself has a pretty long history of cannabis growth and cultivation. So there's a Bell Museum of Natural History, which is in St. Paul. It has an herbarium, and a herbarium is kind of like a pressing of a plant that has a record of where it was collected, when it was collected, what that environment of which it was collected was like. And it kind of gives you a snapshot of how a plant grows in a particular environment at a particular time. So there's specimens that date back to 1867, with a lot of specimens collected pre-World War II effort in 1943. And there's about 176,000 acres cultivated in the upper Midwest during the war effort. And we'll talk about kind of like why this is important much later in the talk. You probably have talked about this before, so I'll go pretty quick, but hemp is largely a legal definition of cannabis sativa L that has less than 0.3% THC. hemp has largely been kind of selected for three major classes of products. You've got your oil seed and grain, you've got your fiber, and you have cannabinoids. Within the fiber, there's the bast fiber. Those are the long fibers that clothing is made out of, that are kind of, you could think of as the bark of the stalk of the cannabis plant. It's that outer fiber, whereas the inner woody hurd is often used in like paper production and like mulch or animal bedding. And some popular cannabinoids that are derived from hemp are CBD, Delta 8, Delta 9 THC, CBDV, THCV, CBG, CBC, and CBN. Yeah, so industrial hemp contains less than 0.3% THC. You can see an image of a fiber crop in the top left or top right there. So it's planted very densely to encourage stock growth, whereas drug cannabis or even cannabinoid hemp, the spacing is much larger because you're trying to encourage a floral yield in production. So yeah. One cool thing about cannabis is that it is largely dioecious. So dioecious means two houses, or that an individual is either male and another individual is either male or female. But there are some fiber varieties were bred to be monoecious, so that means that both the same individual has both male and female flowers. So the image on the left with the pink stigmas are that's a female plant. The image in the middle is a male plant, and that image on the right there it has both male and female flowers there. So cannabinoids, like, why are they important? Where are they made? What are they even? So cannabinoids are, a unique class of compound, largely found in the cannabis species, or traditionally found just in cannabis. I think there are now some lichens and other organisms that produce some cannabinoids, but it's mainly just found in cannabis sativa. It is found predominantly in larger abundance on female plants in structures called trichomes. So these trichomes, that's the image on the top left. So it makes it in that head, the head of the trichome, which is actually non-living cells. So it's like making trichomes or terpenes and cannabinoids in this space, kind of above the plant or on the surface of the plant. Traditionally, plants were largely making THCA or CBDA, though there's been a lot of breeding work for other cannabinoids now, such as THCVA, CBGA, and CBDVA. And it's thought to be largely defense to seed predators. So CBGA is, I guess, colloquially known as like the stem cell cannabinoid, as it can be acted on by an enzyme to either make CBDA, THCA, or CBCA. So CBGA can become multiple different cannabinoids. Traditionally, the major cannabinoids were CBDA and THCA. And there's a ratio trait that our lab worked on, a marker model that to predict if a cannabis plant will be CBD dominant, THC dominant, or intermediate. And this trait seems to be qualitative, so a single trait controls most of that variation, whereas in total cannabinoid abundance appears to be a quantitative trait, or a trait that's controlled by many genes within the genome. So here's kind of a better understanding of this. So if a plant has two non-functional copies of a CBDA synthase, so a synthase is an enzyme that catalyzes a reaction that turns CBGA into CBDA, or CBGA into THCA. So if a plant has two non-functional copies of a CBDA synthase, it produces largely a THC chemotype, or it produces mainly THCA. If a plant has two functional copies, it produces mainly a CBDA chemotype, or produces mainly CBDA. And if it has one functional and one non-functional copy, it produces about an intermediate amount. So I think George had discussed this earlier in his talk, that what they had done prior to my getting there is they had this drug type here on the image on the right, on the left there. So drug type, so that's skunk number one. They inbred it for 5 generations, and they also had a hemp type variety called Carmen, which is now, we'll call it an extant Canadian fiber variety. So they also inbred that for 5 generations. They made a cross to create a true F1, and then they self the cross to create an F2 mapping population. And as you can see, when you self these two inbreds together, they create heterosis or some amount of hybrid vigor. But the more interesting thing that they had found when they were looking at what cannabinoids these plants were producing when you cross a drug type with a hemp type and then self that F1, was that there was about a 1 to 2 to 1 ratio of THC to intermediate to CBD type plants. So very similar to Mendel's peas, and there was what was appearing to be a codominant controlled trait. We're now going to transition to talking about some of the research I worked on at the University of Minnesota. Sorry, I was throwing some slides together last minute. I was unable to remove some of the recordings, but so working with hemp at the University of Minnesota was pretty cool. So when I first started, hemp wasn't yet legal. So I'll talk about kind of what it's like to work under the auspices of DEA and then what it's like to work with hemp. But yeah, we basically just had to fill out a hemp license application. We could grow it anywhere that we reported it on that license application. There was no plant limit. We could immediately put it into the compost area at the University of Minnesota. They gave us a free license. This also opened us up to potential funding opportunities. So we were able to get some MDA grant funding for some of this work. The picture on the left is a picture of me in a plot there in St. Paul when we were doing that grain variety trial, and the images on the right are some images of our greenhouse when we just first started putting plants down there. So what it was like working in the Weiblen lab using under a DA license was we could grow only 40 plants at a time. So when you're doing genetic studies and you need to look at, at least 100 plants or more, it can start to become time consuming. Everything needed to be logged, measured, and weighed. It was stored in an old bake safe, which was like a U of M registrar safe. All of our extra biomass had to be sent to a company that was licensed by the DEA to get burned. We had, it was a minimum security prison style operation. So there's multiple locks and keypads. If I didn't punch my code in fast enough, the cops would show up. Fortunately, that didn't happen for me. The DEA, they viewed DEA viewed the DNA as a controlled substance. So we weren't able to, collaborate with other universities and send DNA to them to do collaborative research. But fortunately for us, University of Minnesota has what they call the UMGC, so the University of Minnesota Genomics Center, which is one of the better in the country. So we kind of lucked out there. Also working under the DEA license. So when I first started, we weren't yet able to collect this feral hemp and do a lot of these feral studies. We were able to update our memorandum of understanding with the DEA to do some feral collections. And so we had to transport around this gun safe. There's a good picture, I don't think I have it in this talk, but of George and JP carting around, putting this.
I'll share it later.
Yeah, putting this big gun safe in a U of M van because they wanted us to have a lockbox just in case this feral cannabis escaped back into its natural habitat of the wild. And yeah, we were unable to secure any grant funding to do any of the drug cannabis research. So it was much harder and more time consuming to work on the drug cannabis side than the hemp side. George had mentioned part of this study, but yeah, we worked a lot with Cystin Wapitin on working on some fiber varieties. Basically doing an economic feasibility study, doing a market analysis, and doing some hemp demo plots. So yeah, there's a picture of I think this is the second year of the study. So we basically, the plants are planted very densely. You end up cutting them down with a sickle bar roller, Basically, when you're going to have like a period with no rain, you'll flip the hemp over once or twice to get it completely dried out, and then it will get baled up. And we got it loaded onto a truck and sold to a company called Basscore. Later on in the study, Sisseton Waupun has, I don't recall the name of this, but George might. They have a Sisseton has a blown plastic bags facility where they mixed polylactic acid pellets with some of the hemp to make these hemp composite plastic bags. I want to say it was like
That's Dakota Western Industries.
Western Industries, yeah. Okay.
Dakota Western, yeah.
And I think currently, I think last time I toured George's greenhouse, they were working on domesticating some of the Sisseton feral cannabis collections to make a fiber variety that Sisseton could market as their own and then control seed price of fiber, because that is one large we'll call it economic roadblock for fiber farmers is controlling that price of seed. The price of seed tends to be, if you were just growing for fiber bales and selling fiber bales, the seed makes that cost prohibitive. So like being able to control your seed price allows you to create more industries and avenues for others. So yeah, we worked on this grain variety trial. We grew 12 different varieties at 5 locations around the state. We basically found, some winners and some losers. X59, CRS1, and Canma were probably the highest yielding grain varieties that we trialed in our study among the 5 locations. Here's a picture of me on the bottom right. This is kind of what cleaning seed looks like. The machine there is an aspirator. So once you've threshed the seed, you run it through this aspirating machine to get the last small bits of chaff and leaf material off of it, and it also blows away the light immature seed and leaves you with a nice clean mature seed product that you could use for planting or selling. So being that our lab was a genetics lab, we're always interested in what's going on genetically in populations of cannabis. One unique thing that we observed is we were analyzing the amount of cannabinoids in some of these plants, and we're noticing some of them are coming up above this 0.3% THC. Some of them, if taken by themselves, would be considered non-compliant hemp. So the question became, why are we seeing this? So we started genotyping some of these plants with our marker assay that we kind of talked about a little bit earlier to determine, our plants, when they produce THC, will they produce CBD, or will they produce an intermediate amount of both? We found, most of the varieties that were studied were intermediate. Or most of the varieties we studied had individuals within these seed populations that were intermediate. We did not observe any drug type chemotypes or individuals. Yeah, so that was like a unique finding that we didn't think we would find in Canadian certified grain varieties, was individuals that contained the ability to produce THC at any amount. So this is some of our feral collection work. I think there's a cooler slide next. We'll talk about that next. But so yeah, so we basically we looked at the same thing in feral individuals from Minnesota, North Dakota, and South Dakota. So we made collections on multiple different populations in these states. And we were interested to see, what chemotypes and what genotypes these individuals had. And what we were finding was, depending on source population, the chemotypes and genotypes could vary significantly. So some populations in Minnesota were predominantly or only hemp, where some had a little bit, were predominantly hemp and had a few individuals that were intermediate, and some populations had a mixture of all 3, drug type, hemp type, and intermediates. One really interesting facet of the study was we found in South Dakota, actually, we found the highest number of drug type individuals we had come across. Though I would not start going and looking for feral hemp this end of the summer, because as you can see here, this is the range of THC content. It ranged from almost nothing to 1.5%. So I think it's going to be a big waste of time if you wanted to collect feral cannabis for its THC content. Though we do find some interesting individuals that, you know, produce almost no cannabinoids to producing almost 10% on this, at least on the CBD side. So yeah. So I'll talk about a little bit. This is kind of a hodgepodge of some science stuff we did at the U of M, but so George had mentioned we had at least in George's video, he had mentioned we had just sequenced the genome of cannabis. So this is what we consider a composite genetic linkage map. So it's like it's basically markers in a space, or I guess marker. How do we say this easily? It's basically markers in physical space that you can then use QTL mapping to place on a chromosome. So it's like we're finding QTLs are areas of the genome that are associated with traits of interest. So we were interested in terpene traits, cannabinoid traits, trichomes, and biomass traits. So we found, yeah, some pretty significant QTL for basically all the trait classes studied. And I think we're also the team. I don't think it's been published yet, but we should be the first team to report a trichome QTL with associated region of the genome. So yeah, a composite genetic linkage map shows the position of known genes or genetic markers relative to each other in terms of recombination frequency. And QTL allows you to kind of visualize this location and have an estimation of the effect of that QTL has on the trait of interest. So we talked at the beginning of the talk about what herbarium specimens are, what an herbarium is. So while at the University of Minnesota, I was able to genotype 166 different plants from herbarium sheets. We used that assay we talked about earlier to see if, you know, can this plant be predicted to produce CBD, THC, or an intermediate amount of both of those chemicals. This assay we used was able to work on even the oldest specimen, which dated back to 1876. And one unusual thing that we found, or one thing that was interesting, was that the presence of the ability to produce THC dated back to 1878. Because previously we had kind of thought that, you know, impure World War II fiber hemp seed was kind of like where this THC producing gene was coming from. But what some of our results were actually showing us was that this gene had been around in these populations a lot longer than we were hypothesizing.
Yeah. Hey, Clement, I might interrupt you there.
Yeah, go ahead.
Might be a good that might be a good handoff to some more talk on the history of the feral and where and how we think that the feral hemp is going to lead the new green revolution.
I'll pass it off to you.
Yeah. But with that in mind, while I pick up that thread, I know that Winona was curious about the bioremediation and that investigation that you did on that. If you want to switch over the screen sharing to me, I could pick up with this feral thread, and maybe you could wrap up with what little you know about the bioremediation. How does that sound?
Sounds good. Yeah.
Okay.
Cool.
Yeah. So there we go. All right. Yeah. So questions for Clement thus far. Feel free to throw them in the chat. I'm going to pick up with this thread on the history of hemp in the upper Midwest. Like, you know, Clement was able to go back, you know, into the collections of the museum and pull out some of these cannabis these plants that are these specimens are over 150 years old now. And he was able to even do the DNA test. I mean, and we know that both the hemp type and the drug type were here way back. And out in the wild, they've been mixing and adapting and evolving and basically making themselves at home in our landscape, uh, free from human, intervention, despite decades of efforts by law enforcement to eradicate this plant. It has continued to thrive, and continued to work with its environment and its neighbors to find a home here, which I just think is really cool. Yes.
Yeah, that's my
Yeah.
question about like, you notice how different plants grow almost together as what I call plant kingdoms or whatever, different communities grow
Yeah.
together. Do
Communities,
you
yeah.
notice that cannabis, like for instance, I notice that cannabis seems to grow in about the same area as nettles. And the other thing is that the plant life that grows tells us about the soil itself. If you watch what grows where, it tells you about the minerals and the
Yeah.
contents of the soil. And so that guides you. So I'm wondering, have you noticed any of that, or tell me what you know about that?
Yeah.
Please, thank you.
Well, you took my best example you've mentioned already my best example that I could give you of a friend of cannabis, nettles. Again, like another plant that enjoys well drained but well irrigated and disturbed soils, all those things. In fact, they're cousins. The nettle family is a close relative of the cannabis family. Right in there with mulberries, the mulberry family. And definitely as indicators of the soil. Yeah, for sure. Cannabis, it's so responsive to the soil conditions. You can almost use the leaf color um, as a measure of the nitrogen levels. Darker green for, you know, lots of available nitrate, and pale yellow for like low nitrate. And you can even see it on the landscape level when we've seen farmers grow, like, they have a field of 40 acres. Let's say down in the in the hollows, there, it's a little bit wetter and there's a little bit more soil organic matter. Up on the hilltops, it's all wind, it's eroded and wind blown. There isn't as much soil organic matter. You'll see yellower, shorter stature plants on the hilltop and darker green down in the hollow.
Yep, that's what I
Um,
would expect.
yeah. So
Because
it's
the up top around here is more clayey, and
Mhm.
the lower bottom field is where naturally you'll find it down in the hollows.
And where are you at?
Well, I'm in Northeast Iowa, but I grew up on horseback. And so when you're on horseback, you're going so much slower that you
Yeah.
notice things like an entire field. Well, God, we discovered when we were teenagers a whole field of marijuana. We thought we were in heaven,
Oh,
but it was
yeah.
just hemp.
Yeah.
It was just hemp. It was not,
Great.
it had resin and all kinds of stuff, but
Oh,
it
yeah.
wasn't,
No,
it was
I loaded
wild.
up a whole van full of that stuff in high school, and you know nearly
Put it in
ended up in
the
prison
oven.
before I realized it was worthless. Um, yeah. Um, yeah, no, I absolutely. Good point. So yeah, just want to show you I'll show you the distribution of this feral stuff that we've been able to, this picking up on what Clement was just talking about with the specimen records. Um, let's see here. Yeah. Um, so whoop, um, I don't know why it says secure there. Anyway, these little green dots, these are museum records from uh all across the United States, that over the last 50 years where the plant's been collected. These aren't all the records, but the density of the dots gives you a pretty good idea of where feral cannabis has thrived. You'll see a lot of the dots follow the Mississippi River Valley down through the border of Iowa, Illinois, um, um, and Missouri. You got a huge like northern Nebraska, Platte River and whatnot. It's just thick. And then of course Minnesota River Valley. It's like a whole diagonal line of dots here from Lake Traverse on down to Bedote. Yeah. So this is the historic distribution, and we got in our region here a lot of good material to work with. We've been going around with a colleague who's in Madison. She's got the red dots for the Badger State, and we got the gold dots here for the Gophers. You can see we've found quite a few of these populations that Clement was just talking about. The backstory to how we were able to do this feral work, kind of funny. Backstory here. If any of you are familiar with Dr. Bronner's magic soap, it's they're night. It's good. It's good stuff. Oops, what have I done here? I popped out of screen sharing. Oh dear. Sorry about that. Dr. Bronner encouraged us to start working on ditchweed. And this was great because it's been almost impossible to get any company or any federal government agency to support our work. And this was back in 2013 before there was a farm bill. And the CEO, not the chief executive officer, but the cosmic engagement officer, David Bronner, gave us a small award to go out and start working on ditchweed. And I love running this sponsored project agreement by the regents of the University of Minnesota, that we have this like contract with All One God Faith Incorporated, and the university was not exactly accustomed to accepting research awards from such organizations, but it was really fun. We got the money and we started to work on this, and it took literally 2 years and a couple of lawsuits to finally get approval of the protocol for going out and collecting ditchweed. Clement alluded to this, how we negotiated this agreement, very strict terms under which we'd go out and get the ditchweed and then bring it back to the laboratory. And among them was using this gun safe, this lockbox for transporting the hemp back to the lab. We've since gone on to find where we can find big stands of it, where we've been able to collect enough of the wild seed to start working with it again. It's been 70 years of adapting on its own in the environment, probably descended from some of those early introductions of fiber hemp throughout the Midwest. But it's changed and evolved. And how has it changed and how has it evolved relative to the commercial hemp seed that one can buy today? That's what we've been studying. And as Clement mentioned, the wild populations are diverse genetically and in terms of their chemistry and their form and everything else. And we've found that about 10% of the ditchweed in our region, 10% of the individual plants are not of the hemp type. They're not CBD type plants. They're either intermediate or their THC type. So part of our breeding effort in the lab has been we take this wild seed, we grow it up in the greenhouse, and then we test it, select it. We choose plants to carry forward that are of the CBD type, so that we're compliant with the law. But we also don't want to lose that tremendous genetic diversity that exists in the wild. And this slide illustrates it really well, where these plants shown here, they're all siblings. They're siblings from the same family, grown out under uniform conditions. And some individuals are short and squat, like a hashish producing plant. Some individuals are incredibly tall and leggy. Some are very highly branched. Some are Christmas tree-like. Some are almost just linear and viney. What this wild natural genetic variation gives cannabis is a lot of resilience. No matter what the environment or humans throw at it, chances are there are some individuals there that can thrive. So with our program to work with the ditchweed and create new native hemp varieties. We have a project, as Clement mentioned, with Sisseton, Wapiti, and Oyate, but we also have a project here in Minnesota with the Anishinaabe Agriculture Institute, and that's the one I want to focus on. Our colleague Tom Michaels in horticulture has collected and selected these ditchweed lines while trying to maintain some of the genetic diversity. And we're growing this stuff in the greenhouse, collecting seed from it after we've tested to make sure that it's drug-free, and then we can bring it out to the farm. And so here's Tom Michaels pictured with my research associate JP Wenger in an experimental stand of the ditchweed here. What we've been able to do with Tom, he's into what's called open source seed sharing. This is the kind of arrangement that you have with groups like Seed Savers in Iowa. What we've done is negotiate between Anishinaabe Agriculture Institute and the university a non-proprietary and equitable seed sharing agreement, whereby whatever we plant at AAI, AAI reserves the rights to half the seed, and we reserve the rights to the other half. I'm speaking on behalf of the university, and each party can do whatever it wants with that seed and can't interfere with the other party. And so what we're hoping here is that out of this foundation breeding seed, we will be able to develop sufficient quality and quantity to start providing a native sovereign produced and adapted hemp variety. Here's some pictures from recent seasons. Just look at the difference in color between Canadian commercial hemp seed and Minnesota wild hemp. The pale bleached coloration of the commercial hemp and this darker color here, one of the things we think might be going on is that the size and pigment of the seed is selected in nature. If you're growing hemp for grain, you want big seeds, and that's what we got over here on the left. But if you're just a wild cannabis plant trying to survive, you might it might make sense to invest less in each seed and invest in a greater quantity of smaller seeds to maximize your chances of doing well in the environment. And we see that trend in a lot of cases of wild versus domesticated plants. Coloration, blending into your background. This darker camouflage coloration, harder for the birds to pick out the seed on the ground. That's the idea behind that. Then, very important, germination. If you're in the seed business, you're required to produce results on what percentage of your seed germinates. And the higher, the better. And it's supposed to germinate all at once. You add water and soil and sunlight, and boom, it's supposed to grow. What we see in the native wild hemp is germination is low under ideal conditions. It's low at first, but it keeps on coming. The germination is spread out over time, and the seed germinates in wave after wave after wave throughout the early part of the season. And it survives cold spring temperatures and then more germinates later. The commercial seed, you throw it into the ground in suboptimal conditions, it won't even come up. And so there's that resilience again in diversity that we think may have advantages moving forward. The immediate germination rate may not be so high, but the seed may do well, as it did comparatively last year on the year before under drought conditions. So I'm gonna wrap up here with just showing you some data comparing Canadian commercial hemp to the Minnesota feral hemp at Winona's farm 2 seasons ago. And what you see is that the plants were at relatively similar density in these two side-by-side plots, but the Minnesota feral was 50% taller. The yield of stalks per acre was 100% greater compared to the commercial. The seed yield in pounds per acre was double, and the biomass yield was 50% greater. And so here's some actual data evidence suggesting that, in fact, some of this locally adapted hemp might even do better here than Canadian commercial hemp. And that's what we want to continue working on. Some of the folks who participated in those experiments, Winona and Don, of course, Jerry Lee Chilton, who's directing AAI at the moment, and Darren, who's an excellent guy to have with in the field. I think I'm gonna stop sharing there and take any questions about the feral hemp that you may have.
It makes me so excited. I collected hemp seed last summer from it was like a beautiful specimen. It was like 10 feet tall. It wasn't marijuana. It was hemp with really good, so yeah.
Yeah. Well, you're going to plan it out? You're going to give it a try?
Yeah, you reminded me. It's sitting right over there in a bag. I kind of forgotten about it, but yeah, it's time to plant.
Well, the key thing is, if you want to sort of work with that and maintain the integrity of that, you want to plant it away from other varieties. And so, like in our little experiment there where we were growing the two side by side, we don't actually keep, they can cross-pollinate. And we don't want, we want to keep them genetically isolated for the time being. So we're not going to collect the seed from the experimental plot. Instead, we've got a separate seed nursery where we're growing the plant we're growing the feral by itself in isolation, trying to increase the quantity of seed we have. And it's a slow process. You know, you start with 2 pounds of seed. You hope that you can plant that out and maybe harvest 20 pounds from that. You plant 20 pounds out, you're hoping you can harvest 200 pounds, and then from there to 2,000. And it's only when you get to 2,000 that you're able to do, you know, a 40-acre field and hope to generate, you know, tons of the stuff and really go into business, really do something with it, turn it into a product. And
Right.
that's kind of where we want to get to. Yeah, I see that we've had some tech difficulties with Winona's question, or with Winona's connection, but Esther's come back on to just let us know about that. Yeah, so keep them coming if you got any other questions.
Yeah, we lost her earlier, and for some reason my video isn't working, so
Yeah.
but I was able to listen to the whole entire thing and I really learned a lot. So I just want to say thank you both for presenting today. Thanks, Clement, for coming in last minute. Sorry, scheduling is a little, we've had to adapt to everybody. We just want to make sure everyone, we're not taking up your whole day, your whole schedule, because we know you both have work too as well. But
Oh, sure
yeah,
thing.
that was awesome. I didn't know. I think I've heard you mention that before about how much better the Minnesota Feral performed, but that's the first time I saw that slide, or maybe that's the first time I've seen that slide and it really sunk in.
Mhm.
I was like, I can't wait. I'm gonna pull this up and show it to Jerry Lee too, because yeah, he had mentioned last year it was so dry. Our area was in a severe drought, and
Yeah.
we paid, AAI paid a lot of money to import a lot of seeds, and some of them,
Yes.
the
But
germination rate wasn't as great as we had hoped, and it would be awesome someday where we could just plant, 100 acres of Minnesota Feral, and it just is years and years and years of work, and you could see how much greater it outperforms the imported varieties, which probably perform great like in their region. We try to buy
Mhm.
things that are, in the kind of like the latitude, longitude, like
Mhm.
Ukraine,
But
Russia,
yeah.
northern China and Asia is kind of similar, like Minnesota, where we are in Minnesota is kind of comparable in weather to like Siberia. So like
Yeah.
the whole thought is like, well, if it could survive, 40, 50 below zero or whatever, it could survive here too.
The
So
big thing, even more than the region, Esther, the big thing here is the practices and the people and
yeah.
it's like, yeah, you can take that commercial seed, but you got to be using chemicals, pesticides, fertilizers,
Right.
herbicide
Mhm.
and then it'll outperform the feral just the way, you know, corn from Monsanto is going to outperform,
Yeah,
Indian corn any day, yield per
right. But
acre.
which one's better? Yeah,
Well, which one's
it's all,
better and which one does better
yeah,
with healthier farming practices, right? We
Right.
right.
think
But it
we
depends.
think the feral is going to perform better with healthy farming practices.
Hmm. Yep.
And we want to prove it and increase it and hopefully grow the community of people who want to use those practices.
Right. I
Yeah.
just wanted to say I want to direct this kind of towards Clemon before he goes. Like part of what he said there was love is an ingredient. And I feel like that is so important. Like so I grew up Creole, trapping, hunting, gathering, small farm and garden, lots of barter and sharing of labor. You did communal labor together, right? And when we look at hemp, one of the big questions is what is it that we value? And so I feel when we're sort of kind of pushing towards the industrial and the large scale. For one thing, in the bioregion in which I live, large scale is not applicable because I'm right about 5 miles from Seed Savers. We live in a very hilly, hollows everywhere, right? That's what it's all like. And so we have small fields, but they can be done like scying. Oh man, my grandfather used to scy the entire alfalfa field, right? Scying is beautiful work. And when I plowed the lower bottom field, we did it with 3 little Haflinger workhorses, right? That the neighbor brought over. And the amount of social capital that was engendered simply by having horses plow the field rather than one person on a tractor, which is the other thing. When I grew up, nobody bought equipment by themselves. The entire community. Now, it has so changed. Okay, it's all corn and soybeans and chemical plowing right up to the riverbanks and stuff like that. So love is definitely, I think, one of the essential ingredients for why to get into this. Which brings me to like doing a research thing with the local Amish farmers would be so interesting because they also have a culture of shared labor and tools and hand tools. And it would be interesting to develop a whole thing that was about small scale agriculture and augmenting small scale farmers. That's all. Thank you.
Thank you.
Sorry.
Cool. Well, that's a great note to end on.
Yeah, that was awesome. Well,
Well, I think-
everybody, any other questions? Oh, go ahead, Katie. Yep.
So just one quick thing, maybe it's more of a comment than a question, but in gathering the ditchweed and experimenting with them, they would have a very unique microbiome on them that would express differently if that were in communication with the microbiome in the ditch versus the microbiome in the soil. And so I just wonder, and don't feel like you have to go into a long explanation because I think we might be wrapping up, but I just wonder about how that role of the microbiome is taken into account in looking at their performance over time on the outside as well as inside the ditch weed seeds.
We haven't I don't think we've looked much at all, if at all, at like the mycorrhizome of feral cannabis, but I think it would make a lot of sense why feral cannabis can perform so why some feral cannabis perform so well in such poor environments is likely it does have some of these may form more communal, more what is it called? Mutualistic relationships, where they're providing, some sugars via their roots to microorganisms and are able to somehow select for these mutual relationships with microorganisms. Because I think me and George were up at the Indigenous Foods Conference, and I think a talk after mine was about a guy that looked at these very similar relationships on no input corn varieties.
Yeah.
And what
The
he was
work on corn, yeah, the corn symbionts that have been ignored and actually suppressed by industrial breeding and the seed industry. And he's worked with these varieties that are outside of that. And man, it was mind-blowing what this microbiome was doing for this corn. It was totally amazing. Yeah. We don't know anything about it in cannabis yet.
I tend to see it as like an edge plant. I see that it doesn't necessarily like full sun all the time, or it does very well in a blend, almost on the edge. As long as it has good drainage and good water. I'm done. Such an exciting topic. It's wonderful. Thank you guys so
Thank
much.
you.
I learned so much.
Thank you very much.
Good. Glad to be able to share time with you. Good luck with the rest of the program.
Thanks so much, everybody.
Thank you. Thank you.
Yep. Have a great rest of your week.
Bye-bye.
Bye.
Thank you.
Thank you. Thank you.